Turns out, multiple paths to long life. Here's the thing about living forever. Your body might not want to do it the same way your neighbor's body does. We've spent decades looking for the fountain of youth like it's a single magic formula. One diet, one pill, one perfect thing. But what if the secret to a long life isn't about finding one path? What if it's about the fact that there are completely different ways to age slowly, and they can actually work in totally opposite directions at the genetic level? That's exactly what researchers just discovered, and it completely upends how we should be thinking about longevity. So if aging pathways contradict each other, why do they both add years to your life? Before we get into the molecular weeds here, let's establish why anyone cares about aging at all. For about the last century, we've been treating aging like it's this inevitable, unchangeable thing. You're born, you get old, you get sick. That's the deal. But starting in the 1990s, scientists realized something weird. Aging itself might actually be the root cause of basically every age-related disease. Parkinson's, Alzheimer's, heart disease, cancer, they all get more likely as you get older. So the thinking shifted. Instead of fighting diseases individually, what if you could fight aging itself? What if you could slow down the underlying process that makes all of these diseases more likely in the first place? That's not just elegant, it completely changes the game. This new study, published in May 2026 by a team led by Rudich and Guan at the University of British Columbia, decided to tackle this from a different angle. Instead of asking, what makes things live longer, they asked, what genes do all the different longevity pathways have in common? That's a way more interesting question. The research team here is based at UBC's Aging Research Centre, and they're specifically obsessed with connecting aging biology to neurodegenerative disease. That's important context because the whole motivation for this work comes from something they've already shown, that you can make animal brains more resistant to diseases like Parkinson's by targeting aging pathways. Rudich and his colleagues wanted to know if they could figure out the deeper mechanics of aging well enough to uncover brand new drug targets. When you're trying to understand something as complex as aging, you don't just guess, you go systematic. Here's what they did. They took 9 different strains of C. elegans. These are tiny roundworms, about 1 millimeter long, that age in weeks instead of years and have been the workhorse of aging research for decades. Each of these 9 strains was engineered to have a mutation that makes it live longer through a completely different mechanism. We're talking 7 different longevity pathways here: insulin signaling, dietary restriction mimicry, no germline, bad chemosensors, slower protein synthesis, extra mitochondrial stress, and mild mitochondrial problems. Think of it like this. You've got 7 different ways to rewire a car engine so it runs longer. One version reduces fuel consumption. Another removes the navigation system. Another tweaks the combustion pressure. All of them make the engine last longer, but they're doing it through opposite mechanical changes. The researchers then extracted RNA from these worms. Basically, they looked at which genes were being turned on and off in each strain. They used RNA sequencing, which is basically reading the entire instruction manual of which genes are active right now. Then they compared all 9 strains to each other and to normal worms. Here's what they found, and this is the wild part. Most of these long-lived mutants looked surprisingly similar in terms of gene expression. They were changing the same genes, often in the same direction. But when you looked at all 9 together, a pattern emerged. The worms sorted themselves into 3 completely distinct groups based on their genetic expression profiles. And here's where it gets actually weird. 2 of these groups were doing opposite things at the molecular level. Imagine 2 people who both live to be 110. One does it by maximizing their immune system. The other does it by actually suppressing certain immune responses. Both strategies work, both add decades. But they're pulling opposite levers. When they filtered for genes that were being consistently changed across at least 6 of the 9 mutants, they found 196 genes being turned up and 62 being turned down. The genes going up? They were mostly involved in immune function, defense against stress, and metabolism. The genes going down? They were involved in translation, basically the machinery that reads your genetic code and makes proteins. That's a huge category of cellular activity, and these worms were essentially dialing it down to live longer. Now, before we get too excited about aging genes, let's ground this in reality. This is worm biology. C. Elegans are not tiny humans. They have about 20,000 genes compared to our roughly 20,000 genes, but they lack a lot of complexity we have. A nervous system, yes. Bones, organs, an immune system that resembles ours, no. So when we find that reducing translation extends lifespan in worms, does that work in humans? Unknown. Does that mean you should eat less protein? Absolutely not. That's not what this says. What this actually says is that there appear to be fundamental principles of aging that worms and maybe other organisms share. Proof of concept. But here's what matters for your actual life. This research suggests that aging isn't like a simple machine with one critical component that breaks. It's more like a complex system with multiple failure modes, and you can age slowly by addressing any of several different modes. That's actually hope-inducing if you think about it. It means if one approach doesn't work for you, maybe caloric restriction makes you miserable, there might be 5 other approaches that would also work. The fact that they're genetically opposite also suggests that different organisms, different tissues, maybe even different people might need different interventions. The limitations here are real, though. This is foundational biology in a model organism. The next step is figuring out which of these 196 genes actually matter in mammalian aging, then in humans. That's years of work. We don't know if the same genes even extend lifespan in mice, much less in people. We also don't know if you could actually target these genes safely. Messing with immunity sounds good until your immune system crashes. We also don't know which of these genes are just correlates of aging versus actually causing the aging difference. They're linked, but that doesn't mean causation. This research sits right at the intersection of basic aging biology and precision medicine. Most aging research has been trying to find the unified theory of aging, one thing that explains everything. This paper suggests there isn't one. That's actually more complicated and more interesting. It opens up questions like, why do these different pathways exist? Is aging actually a collection of problems that can be solved in multiple ways? And could we potentially combine treatments from different groups to get even more longevity? That's pure speculation at this point, but it's the kind of speculation that generates the next round of experiments. The work also has implications for disease. If you understand that there are multiple ways to slow aging, and if you understand that aging is the main risk factor for neurodegenerative disease, then suddenly you have multiple potential intervention points. You could potentially slow Alzheimer's by boosting immunity in one patient and reducing translation in another, depending on what their biology looks like. That's personalized medicine at the aging level. That's farther out, but it's the direction this points. Turns out scientists have been busy, and they're not done yet.